So our chapter on brains and behavior begins with a story, as all chapters do, but it begins with a story about New Caledonian crows, Corvus monodoloides. Now these crows, there are many species of crows around the world. I'm not exactly sure how many species. Right here in Arizona, we have the American crow and we have the Chihuahuan crow. But there's a lot of different crows, and crows and ravens are known to be exceptionally intelligent birds. They belong to a larger lineage or family of birds called the Corvidae. That also includes the jays, like the blue jays, and they are also very intelligent. But the New Caledonian crow really takes the prize. It is perhaps the most intelligent of all birds. New Caledonia is a cluster of islands several hundred miles to the east of Australia, and there are many unique species of plants and animals that live on those islands, given their isolation from other parts of the world. So the New Caledonian crow has evolved in isolation, and it has evolved to be highly intelligent. Now, crows in general, ravens might use tools to various capacities, but the New Caledonian crow has shown the capacity to make tools, to invent tools, if you will, out of sticks and leaves and other things in the environment to help them capture prey. So, for example, the New Caledonian crow might collect a stick and take off pieces of the stick to fashion a hook, and then use that hook in the cracks and crevices of trees to capture bugs and insects to eat. Or they might even pull a leaf off of a tree and shape the leaf kind of like a key in a lock to fit inside of a crevice, and then insects might grab onto that leaf with their mandibles. They pull out the leaf and they eat the insects. So this is bona fide, true tool making and using. An experiment by Alex Taylor, a behavioral biologist, was published in 2010, demonstrated the very complex problem-solving skills of the New Caledonian crow. So take a look at the figure on the bottom left of the slide. What Alex and colleagues did is they placed a piece of meat inside of a glass container, like an aquarium, terrarium, so the crow could see through it, see there's a piece of meat that it would want to eat. They then placed a long stick inside of, so here's the meat down here, they then placed a long stick inside of a cage. And in order to get that piece of meat, the crow would have to use the stick, because the crow's beak just simply isn't shaped in a way that it's going to be able to get that piece of meat. Now, in order to get that long stick, the crow would have to use the small stick on a piece of string to get access to the large stick. The crow figured this out. So the experiment was designed with human ingenuity to understand the pieces of the puzzle that needed a multi-step pieces of the puzzle to get the meat. And sure enough, the New Caledonian crow figured it out, pulled up the string to get the little stick, then used the little stick in its beak to get the big stick out of the cage. And then once it had the big stick, it used the big stick to get the piece of meat. This level of complex problem solving is really on par with, say, what a 4-year-old human being can do. And we all know 4-year-old human beings can be pretty intelligent, smart problem solvers. So this not only is just an impressive story on its own, but it shows that whereas we used to think that humans were this unique species that's able to solve complex problems, in fact, the ability to do this has evolved in several species. So we're not alone. And behavior is not limited to animals at all. But as you'll see in this chapter, behaviors are also exhibited by single-celled organisms. Behaviors are exhibited by plants. And of course, evolution is central to all of this, which is why we have this chapter in this textbook on evolution, because these kinds of behaviors have evolved. And behavior is, in fact, a phenotype. It's a phenotype that, just like anatomy, is determined in large part, by genetics. So because of that, behaviors can and do evolve. So hence they are a subject of our evolution class, yet another component of biology that we're able to understand through the lens of evolution. So the evolution of the central nervous system in animals, by the way, which we're going to address this in this chapter as well, opened up new opportunities for behavior that other types of organisms never had, like single-celled eukaryotes or plants. So this central nervous system has allowed interactions with members of the same species. So cooperation and cheating, tool making in other species, like the raven, illuminates how our own species may have evolved. And so this brings to the top social interactions, the evolution of social communities and social interactions. So those are the kinds of things we're going to investigate in this chapter. And this fascinating area of biology dealing with behavior is something that we can also understand better through the lens of evolution. And that's what we're going to do here. So to get started here, let's first define what a behavior is. Technically speaking, a behavior is any internally generated response to an external stimulus. Fair enough? So with that kind of a definition, then we can see that behaviors are not just restricted to animals, because there are many different kinds of organisms. In fact, all organisms generate some kind of response, some kind of internally generated response to external stimuli, including prokaryotes, including bacteria, and certainly eukaryotes of various types, including plants. So that's what a behavior is. And while we often think of and just restrict behavior to animals, that's not the case. Now, the next question we want to address here is, can behaviors evolve by natural selection? And the answer is yes, because all three criteria that are necessary for natural selection to occur are satisfied by behaviors. Those criteria were outlined in Chapter 2, and those are good criteria to remember. So first of all, the object of what we're interested in terms of can it evolve by natural selection, in this case behavior, we need to first establish or know whether it can influence the fitness of individuals. And certainly behaviors do influence the fitness of individuals. They often determine whether an individual is going to reproduce or not. So check, that's satisfied. Another criterion for something to be able to evolve by natural selection is that it varies from individual to individual. And that's definitely the case for behavior. And the third criterion is that variation, and I'm sure you remember this, that variation is at least in part due to genetics, to genes. There's got to be at least partially a genetic basis. So it's got to influence the fitness of individuals. It's got to vary from individual to individual. And there has to be variation that is at least partially rooted in genetics. And all three of those criteria are satisfied. So behavior can evolve. But it's not end of chapter. We're finished with our discussion. We're going to see how behavior evolves and cover some of the nuances. So here's an interesting story. Back in 1959, a Russian biologist, Dmitry Belyaev, probably pronouncing it incorrectly, started an experiment with foxes. And he was curious if foxes could be tamed in the same way that dogs have been tamed from wolves. Because indeed, dogs are essentially wolves. Wolves that have been domesticated, wolves that have been tamed. It's only within the past several hundred years, especially, that we have, through artificial selection, created all these different breeds of dogs. But up until a couple thousand years ago, from the evidence we have, there weren't very many breeds, but there were dogs, and they were domesticated, they were tamed. So could we do the same to foxes? And so he started his experiment. Now, first, foxes vary in levels of aggression. So there you go. There's one of those criteria for natural selection, is to have variation from individual to individual. So check, that's satisfied. And certainly that kind of aggression could influence fitness. In fact, we as humans can decide, hey, those foxes that are less aggressive, we're going to allow them to breed, to have fitness. So there you go. And this aggression could be shown through experiments to have a genetic basis. So we'll take a look at that in a moment. So he gave tameness scores to foxes over the period of several months. And these tameness scores were based on criteria, such as whether, first of all, a fox ran away if it was, a human was nearby, or whether it snarled, or whether it approached and got closer to the human. And then, depending on the response, let's say the fox approached, then did the fox bite? Did the fox back away? Or did it tolerate contact when the human tried to touch the fox? Did it bite? Did it back away? Or did it tolerate contact? So these different kinds of reactions, these different behaviors, were scored. And this was the tameness score. Now, breeding experiments showed the heritability of aggressive behavior. And you learned in a previous chapter how this is done. So if you look at our figure on the bottom right of the page, we've got the mid-parent behavior score of aggression. And then we plot that against the offspring behavior over on the y-axis. And if there was a random plot, then with no slope to any line, then we would conclude that there's not any significant heritability. But in fact, you can see that there is a significant slope, positive slope to this line, showing that the mid-parent behavior score is directly related to the offspring's behavior. So therefore, aggressive behavior or tame behavior is heritable. There's a genetic basis. So the next step was to impose differences in fitness by the researchers. So those foxes that were the most tame, that had the highest tameness score, were allowed to reproduce. They were allowed to earn some fitness. And over generations, the average tameness score increased. So this alone showed that foxes could be tamed, that they are similar to wolves in that respect, and that this kind of behavior does respond to evolution. This part of our chapter then goes on to speak specifically to identifying the genes or identifying genes that are responsible for this kind of behavior. So a QTL analysis was conducted, narrowed the genetic search eventually to dozens of genes. I see I have a typo there, it says teens. And also was able to show evidence of recent selection by looking for, footprints of selection, also discussed in an earlier chapter. So we've learned quite a bit about the genetics that determines tameness and these kinds of behaviors. And we have learned that these kinds of behaviors can be molded by natural selection. So in fact, the genetics behind behavior is pretty complex. And that's what the figure here is showing you or representing to you. So let me just explain a little bit about this figure. And again, the main point is to get across what the title of the slide is saying: genes regulate behavior at several levels. And it's pretty complex. So when you're looking at this diagram, there are genes that are common among many different animals. If you look at the top of the diagram, we have the EGR1 gene, song recognition in zebra finches. Going counterclockwise, treatment of queens by fire ants, the GP9 gene, etc. So you can look around this figure and see that specific genes have been identified that control these different behaviors in different animals. But look at the inside of the figure, and you can see solid lines are representing the influence of genes on behavior in a moment-by-moment way. So where the brain is directly connected to that. So if you look at the top of the diagram, social interactions between animals, feedback. Those are individual behaviors, but they feed back to the brain. The brain processes that information, and then a behavior is exhibited in response to that. And then we also have our genome, and the bottom of the diagram is also directly affecting brain function in terms of which genes are expressed and which genes are not expressed. So that's kind of the highway, the conduit in the middle there, showing the influence of genes on behavior kind of moment by moment. But then there's dotted lines that are shown in this figure, and the dotted lines are here. See little dots. And those are showing behaviors that are exhibited over the course of development. So through the development of an embryo, through the development of a young individual. So you can see there, the genome, of course, is involved in the bottom. And again, we have social interactions are interacting with the genome and affecting development. So an example there with birds is many birds' songs are learned very early in their life. And they have to be exposed to the songs of other birds of their species in order to have a narrow window of time to be exposed to learn those songs effectively. So there you go. There's interactions, social interactions, interacting with the genome and influencing development. There's also epigenetic changes that you can see on this diagram. So interactions of multiple genes that are feeding back to the genome. And then finally, we've got dashed lines on this diagram, and those are showing behaviors that are in response to social and the physical environment. So social interactions between organisms and a population are feeding back and influencing evolution and the genome. So the overall conclusion here on the bottom is that it's very challenging to pinpoint the effects of individual genes on behavior because of these complex interactions of many genes interacting with many different environmental stimuli. And so it's a tough call to figure this out, but of course it's something else that we are doing. So while we can see, like the last slide showed, that the genetics of behavior is very complex. There's lots of complex interactions, but we can figure out those networks. It's also being shown here that behaviors can rapidly respond to natural selection. It does not take a long period of time for behaviors to respond. Indeed, in other chapters, we have seen other traits that respond very quickly to natural selection. So just a few examples here to illustrate the point. So the top figure, figure A up here, is representing that mice that spontaneously ran longer on a wheel in a lab were selected. And after 30 generations, mice were running 3 times more on the wheel than the control mice. So selected lines of mice running more on the wheel were allowed to breed, have higher fitness, versus controls where there was no selection. And we can see that behavior rapidly evolved in just, well, we can see it particularly was evolving quickly, like in the first 20 generations. And then it sort of stabilized out. The middle figure, figure B, is showing how mice were selected for aggression, making more attacks on other mice. And again, those mice that were selected for aggression, you can see over generations, not very many, aggression increased. And that's in comparison, importantly, to controls. And then the final example here on the bottom figure, figure C, is with great tits, Paris major, little bird in Europe. And these birds were selected for their willingness or their reluctance to explore, which exploration is very important for these guys to find new food sources. So a line that was selected for willingness to explore, indeed, over just, you look at the figure, 4 generations, showed significant evolution in that behavior, versus those birds that were selected for a reluctance to explore, indeed also showed evolution in that direction. So here are 3 pretty dramatic examples you can carry away with you that illustrate that behavior can respond very rapidly to natural selection. So the last slide showed 3 examples of rapid behavioral evolution in controlled laboratory settings. Well, what about evolution of behavior in the wild? Well, here's a couple examples. Juvenile Sticklebacks, which are these fish depicted over on the figure in the left there. They're not very big, few inches long. They're found in both freshwater lakes and in marine saltwater environments. So they're fairly adaptable to different salinities. And sticklebacks have been the focus of research of numerous topics involving behavior and evolution. So these sticklebacks in the Arctic have only recently evolved to be living in freshwater lakes since the end of the last ice age. And so these freshwater lakes are no more than 200,000 years old. And these sticklebacks have shown dramatic evidence of behavioral evolution between these freshwater lakes and their marine environments. In fact, the fish in freshwater lakes are less social and they are more aggressive compared to their marine relatives. You can look at the graph here is showing several comparisons. So stickleback population, marine versus freshwater, and then on the y-axis it's plotting out a trait. So for the first figure, time spent with a group, so social behavior. And you can see that in a marine environment, sticklebacks are much more social than in a freshwater environment. If you look at the middle figure, marine versus freshwater, you can see what's plotted, aggressive behavior. So freshwater sticklebacks are, on average, much more aggressive than their marine counterparts. And finally, the third graph, marine versus freshwater, number of fish that schooled, that were in a school. And you can see there's more schooling in the marine environment than freshwater. So there's some three distinctive differences in behavior. And what we can sort of find here is that in the ocean environment, there are several significant natural predators. Whereas in the freshwater environments, these predators are absent. So it seems that this relaxation of the selective pressure of predators is what switched the behavioral evolution in the freshwater environments. So it's less beneficial to be spending time in groups. It's more beneficial to just be individually on your own. Hence, aggressive behavior is selected because that is something that's going to lead to more isolated individual lifestyle. And of course, fewer individuals in the schools. So there's an example in the wild of some relatively, in the big scheme of things, rapid evolution of behavior within one species, depending on whether the populations are living in a marine environment or a freshwater environment. Now, the other example given here is with the mouse that we dealt with in an earlier chapter in terms of the evolution of their coat color, Peromyscus, the old-field mice. And most mice in the genus Peromyscus dig a relatively short tunnel underground where they live, escape predators, whatever. Old-field mice, the specific species we've dealt with already, dig a long tunnel with a side branch. So if you look at the bottom figure, it shows you there's the entrance to the tunnel, and it's a pretty long tunnel. Here's where the nest is in the tunnel, where the female takes care of her young. And then she has an escape tunnel up here that is constructed very close to the surface of the ground, so that if a potential predator does enter her tunnel, whether it's a snake or maybe some other small mammal or whatever, then she's able to rapidly dig out of this escape tunnel and get out. Other mice don't do this. So to investigate this behavior and look for the genetic basis of this behavior, old-field mice were mated with deer mice, who are very closely related, and they're able to form hybrids. And the F2 hybrid showed these three variant behaviors in terms of tunnel building. So once those behaviors were identified, then QTL analysis found three loci with additive effects. And each one of the alleles for these loci led to a 3 centimeter longer tunnel per allele. So that's a pretty interesting specific genetic basis there for this behavior of building longer tunnels. So now these researchers are investigating how environments may have driven this evolution, because that's an interesting question in itself. What is it that caused the old-field mice to evolve in this direction? But nonetheless, these are two excellent examples of the evolution of behavior in the wild, and these are relatively recent evolutionary events. Now, we can't discuss the evolution of behavior without bringing up Nico Tinbergen. If you've taken any psychology or any kind of behavior class, then you've almost certainly heard of Nico Tinbergen. He is, quote, the father of behavioral ecology and that research field of behavioral ecology. And his roadway, the road that he laid out in terms of how to investigate and study behaviors, boils down to these 4 questions. And these questions, well, any question that you ask about behavior in terms of research can be classified into one of 4 types of questions. And so this is super helpful for anyone who is getting into research on behavior. So the first question is, how does the behavior work? So that is a physiology-based question. That is a question that's getting at, what's the chemistry and what's the genetics, what's the control behind the behavior. So that's one aspect to understanding behavior, and that's one category of question. The second category of question is, how does the behavior develop within the lifetime of an organism? So within you or me, once we were born and we began to develop and grow, how do certain behaviors in humans develop? The third question is that we may ask. That doesn't mean you have to ask this as a researcher, but this is just saying these are the 4 categories of questions that we ask in behavioral research. What is the adaptive value of a behavior? How does it benefit an organism to exhibit a certain behavior? And then the fourth kind of question, the 4th category is, how did it evolve? Now, questions 1 and 2 are referred to as proximate level questions. And questions 3 and 4, the second 2 sets of questions, are ultimate level questions. So what do those terms mean? Proximate. Proximate level is mechanism level within an organism. What causes something to happen to an organism within its lifetime? Those are proximate mechanisms. How does it work? How does it develop? Those are proximate questions. Ultimate questions are evolutionary questions. Where did this behavior ultimately come from? And so the adaptive value is an ultimate question. That is something that would have come about through the process of natural selection. So that is an evolutionary question anytime we're asking about adaptive value. And how did it evolve? Okay, that's having to look at evolutionary pattern, at a phylogenetic pattern. So a phylogenetic analysis needs to be conducted to track or trace the evolution of a behavior to understand how it evolved, the pathway it took to evolve. So proximate and ultimate questions, these 4 questions are how we frame our research in behavior. So the figure, I've totally ignored this figure so far on the slide, besides Nico Tinbergen's picture up on the top. And so figure A on the top is basically coinciding with question number 1 over on the left. How does it work? So it's just showing that neural circuits and the genes that control those neural circuits, like in a bird, like in a songbird here, that's going to be a proximate question, a mechanism question of how does it work. Figure B in the upper right, how does it develop? Okay, well, bird songs like the one depicted here are learned in life, especially during a sensitive window of development. And so what figure B is showing you is the sound patterns produced by a wild chaffinch song, a chaffinch that's grown in the wild, versus the song patterns of one reared in isolation. And the one in isolation is not nearly as complex. It doesn't have the nuances of the song that it otherwise would learn if it was in the environment, in the wild. So that's a developmental, proximate question. Figure C, what's its adaptive value? So there's a photo of a bird singing, if you will, vocalizing. And so why is it doing that? What's the benefit to vocalizing? What's the benefit of the bird's song? So that's an adaptive ultimate question, a why question. And then finally, how did it evolve? So there is a phylogeny of orioles, type of bird, and it's mapping out clicks that are found in the songs of orioles. And so you can see in pink lineages without clicks in their songs. Green is lineages with clicks in the song. And otherwise gray is evidence for click evolution is equivocal. So we can see through the phylogeny the pattern of how the song evolved. And if we combine that with studies of the adaptive value of the song, then we can perhaps get a clear picture of song evolution. So this is really crazy because, like I said at the beginning of the chapter, behavior is something that's not just restricted to animals, but behavior is any internally generated response to an external stimulus. And as I said, and as you may have read, that means even prokaryotes and plants exhibit behavior. So animals are not the only organisms that show behavioral evolution. And one really cool dramatic example that's shown here is with a unicellular slime mold, Dictyostelium discoides. And it's a single-celled organism that creeps around in the soil like an amoeba and is a soil predator and takes in bacteria by endocytosis, phagocytosis, and eats them. Now, interestingly, with these unicellular slime molds, when food begins to dwindle in the environment, food resources are disappearing, then things get kind of sketchy for a unicellular slime mold. It's going to be more difficult to find food. So they start releasing extracellular signals. And these extracellular signals, and take a look at the figure in the bottom right of the diagram that's kind of showing what's going on here. So because of around 1 o'clock in the diagram, it says feeding amoebas. There they are. Then they're starving. Okay, so they start sending out these chemical extracellular signals, and it brings different slime molds together. So you have kind of an army of unicellular slime molds that are coming from different areas, and they're congregating. And they group together and aggregate into a large mass of cells that starts moving around like a slug. So we have this species of organism that's gone from being unicellular to being multicellular. This isn't over generations. This is within the lifetime of these unicellular organisms. They are now aggregated and massed together, forming this slug-like creature. Now, this slug-like creature made up of all these individual cells moves faster. So it's able to crawl and creep and burrow through the soil and find food faster and consume it. Now, there's different kinds of cells in this slug-like entity. Cells called sentinel cells move up to the front of this slug-like mass, and they sweep up bacteria and engulf them. They fall behind and they die. They're like sacrificing themselves. Eventually, the slug stops, and the leading cells, the cells up in the front of the slug, form a stalk. And you can see that down around 5 o'clock to 6 o'clock down and 7 o'clock on the figure. So those leading cells form a stalk. So here comes the stalk, and they die. Now other cells migrate to the top of the stalk, and they form a bulb. So here's the bulb up here at about 9 o'clock on the figure. And the cells in that bulb release spores, and the spores are dispersed into the environment and develop into new amoebas. So remember, we have multiple single cells that are coming from different directions. They have different genetic backgrounds. environments, different genetic makeups. And so this slug-like creature is moving along. It's made up of different individuals, all working together. And some of these individuals appear to be sacrificing themselves, and only some individuals make it to the top and release spores. Now, researchers discovered a mutation by chance that causes cells to be cheaters. Interesting. And they use this to their advantage to see what would happen if you mix cheater cells in with wild-type regular cells. And the gene that they identified that causes these cells to be cheaters is called the Chta gene. So the Cheetah mutants can't form stalks. So if you just have a slug-like mass that's made of Cheetah mutants, they can't form stalks. They won't form stalks, so they don't reproduce. But when you take some of these Cheetah mutants and you mix them with wild-type slime molds, the stalk forming returns. And the proportion of mutant alleles in spores is higher than in the original population. So these cheater slime molds are making it to the top of the stalk at the expense of all the other wild-type cells, and they are proportionally releasing spores with their genetics more often than the wild-type ones. So if you take one Cheetah mutant and combine it with 1,000 wild-type slime molds, it's on the bottom of our slide here, after 12 generations, 100% of the spores carried that Cheetah mutant. So that is showing that this cheating behavior, in fact, can be perpetuated and can be selected and can evolve in even a creature as simple as a slime mold. And, of course, this figure, like I started off saying with the title, illustrates really importantly that behavior is something that exists even in single-celled organisms that don't even have a brain. So we just looked at an example with unicellular slime molds of how a complex behavior can exist even without a brain, and how that behavior can evolve. Here's another good example of behavior without a brain turning to the world of plants. So plants show a number of different kinds of behaviors. So for example, if one plant is overshadowed by another one that's growing taller, then the one that's being overshadowed, it isn't getting as much light. It speeds its growth up so that it can get the tips of its stems exposed to light and do more photosynthesis and compete more effectively. That's a behavior. It's a response that's internally generated, making the plant speed up its growth, and that response is a reaction to an external stimulus, another plant growing on top of it. Another example of a plant behavior is how roots grow around obstacles. So as roots are growing downwards, responding to gravity, if a root encounters a rock, a large rock, it'll grow around the rock. So that also is a behavior. Or tendrils of vines. As a vine is growing upwards, vertically, say up a tree or up a wall or up a log or whatever, a tree trunk, then the tendrils that the vine puts out follow a circular path as they grow. So they kind of circle around as the tip of the vine is growing upwards. And they do this circular kind of motion until they come into contact with something. And so by doing this circular motion as they're growing, it increases the chances that they're going to come into contact with something. As soon as they come into contact with something, then that tendril wraps around that object and secures itself. That's a behavior. Now, Venus flytraps are probably one of the most amazing examples of plant behavior. So they have 3 or 4 trigger hairs that respond or cause changes in trigger pressure in the cells. And these trigger hairs need to be hit, several of them. An insect can't just hit one trigger hair, but there has to hit a couple of them within a short period of time. And now plants, of course, don't have a nervous system to be able to send signals like animals do, but there are internal chemical changes that take place when these hairs are touched, when there's a stimulus, and those internal changes cause changes in trigger pressure. So a pretty rapid response that causes a venous flytrap to close up. And then, finally, many plants also respond to herbivores, animals that are eating them. So they may start producing more toxins to dissuade animals, herbivores, from eating them. Or plants might release chemicals into the environment when they're being eaten by herbivores, and those chemicals attract predators to the herbivores, thereby helping to protect themselves. So here's a whole array of examples of how plants have no brain, and yet they also exhibit behaviors, responding to the environment with a response due to some kind of external stimulus. All right, so we've just looked at an example from unicellular life from the slime molds. We looked at several examples from plants for behaviors that have evolved. And now we're going to turn our attention to animals. So let's deal with the topic of behavior and the origin of nervous systems, because indeed, with animals, the nervous system is the primary conduit through which behaviors are being expressed. So all life, prokaryotes, different eukaryotes, are exhibiting behaviors through cell communication and changes in gene expression. That's what's going on with everything. In animals, specifically, nerves are sending pulses of electrical changes from one end of the cell to the other end of the cell. And once an electrical impulse gets to the end of a nerve cell, then the signal can be transmitted to the next nerve cell, the one that's connecting to it. So the signals move between neurons or nerve cells at these places called synapses, where the nerves are coming into contact. And neurotransmitters, chemicals, are released from the one nerve cell, and receptors on the other nerve cell pick up these neurotransmitters, and then an electrical signal is sent through the next nerve cell. So if you look at our broader figure of the nerve cells, you can see the synapse. The connection between the nerve cells is right there, and that's how nerve cells are communicating. And they have these arrays of dendrites, these tree-like extensions coming out that are making contact with other nerve cells. So animal brains have about 86 billion neurons in them. And out of these 86 billion neurons, there's about 100 trillion synaptic connections. So a system that is far more complex for internal communication, leading to behaviors, than any plant or any unicellular creature is able to do. So one big question then, of course, is how and when did nervous systems evolve? And that's what we're going to go into next. So, of course, one way we can approach the question of behavior and the origin of nervous systems is by looking at fossil evidence. And, of course, this, to remind you, a few slides ago, talking about proximate and ultimate questions, this would be an ultimate question. How did the nervous system originate or evolve? That's an ultimate question. So going all the way back to our good old Cambrian explosion that occurred beginning around 540 million years ago, essentially, we see this rapid evolution of different animal types, especially bilaterine animals. And we actually have traces of nervous systems recorded in the fossil record. And so we've got one fossil in particular that's represented here on this slide with this tongue-twisting name, Allalocmenius. Try to say that 5 times really fast. That's dated at about 520 million years old. And the nervous system of this creature, it's an easier way to refer to it, is very similar to living relatives, which would be arthropods. So you can see that, figure A is showing you the actual fossil, and then figure B is comparing the nervous system of Allalocmenius to the nervous system of a larval horseshoe crab or to a scorpion, 2 different kinds of arthropods. And you can see remarkable similarity among the 3. So the hypothesis, so there we can see well-developed nervous system in the earliest arthropods showing an apparent homology to the nervous system today. Now, one of the primary hypotheses for the evolution of our nervous system, in terms of talking about the origin of our nervous system, is, and we can see this actually, it's been empirically demonstrated that there are several genes that have been co-opted and used for cell-to-cell communication that were used in single-celled eukaryotes, and these genes have been co-opted and used in animal nervous systems. So genes that were already in existence, already in operation for cell-to-cell communication in unicellular eukaryotes were co-opted and used to build a nervous system. And so we can look at this color coding on the right-hand side of the slide, neuronal proteins, and we can see that some of the neuronal proteins that are used at the synapse between 2 nerve cells to send neurotransmitters and communicate between nerve cells, some of these proteins are rooted in genes of ancient eukaryotes. So all eukaryotes have these genes. Other genes are holozoan origin. So that would be tracing back to choanoflagellates, our closest relatives and animals. Other genes color-coded in light blue are just animal origin, so sponges and jellies and other things. Others are specifically eumetazoan, so that excludes the sponges. And then finally, you can see in this example some of these color-coded proteins, neuronal proteins that are pink, are specific to bilaterian animals or vertebrates. So yes, our nervous system has developed and evolved using genes that already existed in eukaryotes in general, or existed in common with choanoflagellates, and then we built the nervous system from there, and more genes became involved in synapses. So the next question that we're going to look at in the next slide is, did the nervous system evolve in the earliest multicellular animals or in a more derived animal clade? So that's where we're headed next. So to examine this question of, where did the nervous system originally evolve in animals, we got to turn to sponges, because the preponderance of evidence suggests that sponges represent the basal lineage of animals, the earliest branching off lineages of animals. There's been conflicting evidence that suggests alternative hypotheses, but at this point, the overall weight of evidence says it's sponges. And this makes sense for a variety of reasons. Sponges begin life. I mean, here's an image. Here's a photo of a sponge on the bottom left-hand corner. So there's a living adult sponge. But sponges begin life as a larva. It's called a planula larva, and that's shown on the bottom right. There's a microscope image of this planula larva. And there's genes that are expressed in hairs that are on one end of this planula larva. And these genes are also found in neurons of eumetazoans, that is, the other animals besides sponges. So this would suggest that, in fact, there were the genetic basis for making neurons existed in sponges, even though they do not have a nervous system. So we might think of these kinds of cells as protoneurons, as forming the basis to make neurons. These hair-like cells sense the environment and they respond to the environment. So this is a little window that gives us a peek into how the nervous system with neurons could have evolved in eumetazoans, because there already was sort of a protoneuron condition established in sponges for sensing and responding to the environment. Now, these two tasks of sensing the environment and responding to the environment were later divided between specialized neurons that deliver sensory information only, and others that only send out commands for what to do. That's what happens in eumetazoans later on. Now, we just took a look at the last slide at how perhaps the nervous system evolved using the phylogeny, where sponges are the basal lineage, in which case those hairs that are at the front end of the planula larva in a sponge might suggest to us what a sort of protoneuron started out as. But there's another hypothesis for the evolution of animals that places these creatures called ctenophores, as shown on the spectacular photo on the right-hand side of the slide, as the basal lineage of animals. So there's been a lot of conflicting evidence that sometimes shows the sponges as the earliest animals and sometimes the ctenophores. Now, this gives rise to alternative hypotheses for the origins of the animal nervous system. If this is the case, that ctenophores represent the earliest lineage of animals, then this brings us to the conclusion that a nervous system evolved in the common ancestor of all animals, including sponges, but then later was lost in sponges. Okay, so that's if the ctenophores are a basal lineage. But also, it's possible that a nervous system evolved independently in the ctenophores as compared to the eumetazoans. So if ctenophores are the basal lineage of animals, which currently the evidence doesn't quite support that, there's those possibilities. So now, the independent origin of ctenophore nervous system is in fact supported by a couple other facts. For example, ctenophores do not express many of the proteins used by other animals in their nervous system. So this is somewhat suggestive that ctenophores may have evolved their nervous system independently from other animals. Also, ctenophores have many unique neurotransmitters that other animals, other eumetazoans, do not have. So that also is suggestive that ctenophores may have evolved their nervous system independently. So, overall, of course, the nervous system allows animals to gather information and then allow rapid coordinated responses. So clearly, this has been selected and favored in animal lineages and is a highly valuable adaptation that increases fitness. So whether ctenophores share the evolution of the nervous system with other animals, eumetazoans, or ctenophores evolve their nervous system separately and independently, either way, these groups of animals having a nervous system that does this kind of thing is extremely valuable for processing this kind of information and then coordinating responses. Now, if we go, back to looking at the fossil record and what it might show about behavior, trace fossils, those fossils that aren't showing the actual animal, but like tracks on the bottom of the ocean or burrows, they show that some kinds of very simple bilateral animals were grazing on cyanobacteria, just plowing along the bottom and eating them. Then later on, a little bit later on in the timeline for these trace fossils, we start seeing burrowing. So simple animals are burrowing through the substrate of the ocean and acting as predators. A little bit later in the fossil timeline, we see more complex patterns, like coordinated, directed behaviors going up or down and or in different directions to search for prey. So what that is illustrating through these trace fossils is it suggests that nervous systems were evolving and behaviors were becoming more complex once the nervous system was established in bilateral animals. So now we're going to enter into the realm of innate and learned behaviors, distinguishing the two and looking at how the ability to learn can evolve. So some to many animals, behaviors, depending on the kind of animal that we're talking about, whether it's a minority or maybe majority, are innate. And with innate behaviors, not all environmental information is evaluated and analyzed, but the behavior is more triggered in terms of some kind of stimulus, immediately causing some kind of response, which is the behavior. One example that comes to my mind right away when I think of an innate behavior is physically touching the palm of a newborn human baby's hand with your finger. And the innate response, not learned at all, ingrained in the genetics to respond to that stimulus of something touching their palm, is to immediately grasp the finger. So that's a primate response. That's a genetically determined response that's found in our other primate relatives when they also are infants. It's a very adaptive behavior for grasping in an arboreal environment. So that's a good example of an innate behavior. Now, Nico Tinbergen's research included some very pioneering research on herring gulls, which there's a herring gull depicted down on the bottom of the page. And what his research described was that as a parent gull approaches its nest, then the chicks that are in the nest begin to peck at the bill of the parent. And in response to the chicks pecking at the bill, the parent regurgitates its food, throws it up, and the chicks eat it. So Tinbergen did some controlled experiments where he presented chicks with variable types of heads, which you can see in the figure on the bottom. One of the heads depicted a herring gull very accurately with a red spot on the tip of the bill. Another head had a spot, but it was black. Another one was blue. Another one was white. And finally, one herring gull head had no spots on the beak. So he presented these models to chicks, and the results are shown down in that graph. So every single time, the red spot was a completely sufficient stimulus to elicit the begging response in chicks. From the very first time that they saw that red spot, they exhibited that behavior. So there was no learning involved at all, but it's completely innate. And then as you can see on the graph, a black spot elicited a relative pecking response of 86%, and then down from there with the blue and white. So clearly this is an excellent example of an innate behavior. The herringold chicks are born with this particular response. They do not have to learn. And, this whole bill pecking behavior is a property that emerges from the totality of development of the bird. In other words, there's not a single gene that's causing this, but there are multiple genes that are involved with the development of the nervous system in these birds. And it's a complex, it's a relatively simple behavior, but it has a complex genetic background in terms of development. Right, so let's turn our behavior to learned behavior with this example from the sea slug, Aplysia. So these sea slugs, like any sea slug, breathes with gills, and these guys have a special siphon that you can see at the top of the animal here, where they draw water in through the siphon and then over the gills for gas exchange. So the advantage of doing this is that the gills are protected inside the body, and that this specialized tube is able to deliver water over the gills without exposing the gills to damage. Now, if researchers took like a little bristle or a little hair and gently touched the siphon, then you'd get a weak retraction of the siphon as sort of a defensive protective behavior. Very weak, not very strong. So you could interpret that to say the sea slug did not interpret that gentle touch as a major threat. Now, if an electric shock was applied to the tail of one of these sea slugs, then there would be a very strong response, and the siphon would be retracted to a significant degree. But once that tail was shocked, then that behavior did not last very long. After a relatively short period of time, the siphon would go back up, and it wouldn't respond to touch anymore. Now, if the tail of the sea slug was shocked repeatedly, then the behavior lasted for weeks and was a little more ingrained as a learned behavior, a learned response. So then researchers went in, and here's where it gets really interesting, I think, went in and looked at the actual nervous system of the sea slug to see what happened to the nervous system after these stimuli, to the electric shock, etc. And what they saw was the nervous system was rewired with changes to synapses, with changes to some of the dendritic connections. Some of the synapses had become stronger and others had become weaker. And this is referred to as synaptic plasticity. And this is what is behind, this is what this example shows us, learned behaviors that become long-term, that become more ingrained in an animal. It has to do with the repeated stimuli or repeated stimulus initiating new synaptic connections between nerves that then make the response to that stimulus more long-term and ingrained. And from an evolutionary perspective, looking at the end of this synaptic plasticity is adaptive. Because, for example, if a predator tries to grab a prey, like the sea slug, and that happens several times, then new synaptic connections will be made, and the behavior will become more of a long-term behavior that is, in fact, adaptive. So what we can really come away with from this is that learning, the ability to learn, and learning enables optimization of innate behaviors. in a changing environment. So for the sea slug, it's an innate behavior is to retract the siphon and protect the gills. So that's innate. But depending on the environment that the sea slug is living in, that behavior can be optimized for the specific circumstances of that environment, and thereby maximizing the potential for fitness, higher fitness. So this is really interesting, I think, showing that learned behavior involves a rewiring of the nervous system, and the ability to do this, the fact that this has evolved, this ability to do this, is an adaptive behavior, adaptive response. All right, so here we can see a great example, controlled experiment conducted in the lab with fruit flies that shows how rapidly the ability to learn can evolve. So what researchers did was they took orange jelly and they took pineapple jelly, which the flies can taste the difference, and they spiked the either one or the other jelly with quinine, which is a chemical that doesn't have any odor, but it tastes horrible. The flies hate it. So the flies learned to avoid the jelly that had the quinine. And researchers observed that faster learning flies that learned in a shorter period of time which jelly had the quinine, produced offspring that were also faster learning flies. So that helps to demonstrate this heritable component to this ability to learn, fast or slow. And of course, that's one of the components to being subject to natural selection. Remember, is that there's variation and that the variation has a genetic component. So experimenters set up a controlled experiment where they gave the flies 3 hours to learn which jelly was spiked. So they put quinine in, say, the orange jelly, and then give the flies a choice of the quinine orange jelly or normal pineapple jelly. And after 3 hours, they allowed the flies to mate, and females laid their eggs. The researchers then collected eggs from the quinine-free dish. So those females that selected the dish with no quinine, tasted good to them, those are the eggs that the researchers gathered up and reared them. Now in the next generation, they switched the quinine. So if they started off in the first generation, say spiking the orange jelly, then in the second generation, they spiked the pineapple jelly. And then back and forth into the third generation, etc. And they predicted that this procedure, this experimental design, would cause the evolution of learning rather than the instinct to favor a flavor. Because if they just spiked the same flavor every single generation, then in fact the flies may have been just adapting to that flavor. So they were curious about the evolution of learning, so switching it back and forth could demonstrate exactly what's going on behind that. So they did this for 15 generations, and then they compared the controls to the experimental line. And indeed, the selected line at the end of 15 generations needed less than 1 hour to learn. And remember, they began the experiment by giving the flies 3 hours to learn. So this shows that indeed the ability to learn faster has a genetic component, and hence the population can evolve if there is a selective benefit, if it's adaptive to learn faster. So what this also shows us, because there was decreased longevity for those flies that learn faster, they didn't live as long. So there's a trade-off. We've seen this kind of thing before, where a certain trait that carries a benefit might then have negative consequences in other areas, like when we looked at antagonistic pleiotropy. So the benefits of one particular adaptation must outweigh the costs. So in order for faster learning to evolve, that has to outweigh the costs of not living as long. That's the only way faster learning would evolve in the environment. So, understandably, there's limits to how fast flies can learn by evolution, because at some point they just might not live long enough to effectively reproduce. But this kind of trade-off and this kind of ability to switch back and forth, again, also points out that this is a suitable adaptation for unpredictable environments when things are switching back and forth. So we just got out of examining innate behavior and how innate behavior can be adaptive and of course can evolve, but also learned behaviors, that the ability to learn can evolve, and that learning involves the formation of new synaptic connections. So now we're shifting gears again. This is a new section in our textbook titled The Vertebrate Brain. And so we're going to examine how the vertebrate brain has evolved, leading up to a look at ourselves. So, in looking at the history of vertebrates, the fossil history, this creature that's depicted in the figure on the upper right is called Hykuichthyes, and it lived about 530 million years ago. So that is during the Cambrian explosion, and it is one of several different early vertebrates that we have recorded in the fossil record, for which there's no equivalent living today. It was not very big, so when vertebrates were getting started, this guy was only 3 centimeters long, and it did show characteristics, some character states that are uniquely vertebrate. So we're confident that it was a very, very early kind of vertebrate. It did have its spinal cord surrounded by vertebrae, so of course that's a good indicator. It had pharyngeal bars and slits with gills. You should remember that animals like lancelets, cephalochordates, and urochordates don't have gills, but they do have pharyngeal bars and slits that they use for suspension feeding. These animals did have sense organs such as eyes and nostrils, which the outgroups to vertebrates don't have. And they seem to have a cartilage cranium surrounding their brain. So pretty distinctively and obviously an early vertebrate. Over the next 100 million years, fishes, different species, diversified into larger predators with longer neurons. So to become larger, you're going to have longer nerve cells, longer neurons. And going along with that was the evolution of the myelin sheath that surrounds our nerve cells. And you can go a couple slides back to see a PowerPoint slide that shows nerve cells synapsing with the dendrites. And that figure shows a nerve cell with a myelin sheath. And what the myelin sheath does is it insulates the nerve cell and helps to improve electrical signaling. Kind of like putting a rubber plastic insulation around copper wires helps to improve the electrical signaling. So with myelin sheaths and then motor neurons coming into existence, that allowed complex muscular control. And this, of course, led to more complex behaviors. So there's a glimpse at early vertebrates and what they were like, and some of the early evolution of the nervous system. And now let's take a close look at vertebrate brains and how they evolved in response. So if we want to look at how the vertebrate brain evolved, that's an ultimate question. That's an evolutionary question. And for that, we need to take a phylogenetic perspective and look at different vertebrates at different points in time and construct a phylogenetic tree, and then map out on that phylogenetic tree the brains of the different vertebrates so that we can see how vertebrate brain evolution unfolded. That's how we would do this. So what we can see when we do that, check out the figure in the lower left, is that there are common brain partitions, common brain structures to all living vertebrates. So the lamprey is used as an example of one of the earliest lineages of vertebrates to branch off, probably about 580 million years ago. And then we have sharks branching off, bony fish, and of course the tetrapods, amphibians, mammals, reptiles, and birds. And we've got brains here that are color-coded, so you can see that brains have common features like a cerebrum, a cerebellum, optic tectum, and olfactory bulb. And these different regions, of course, are specialized for processing different sensory information and the animal responding. So, what we can also see, though, is various lineages of vertebrates have specialized brain regions, different brain regions that are specialized. So if you look at the sharks, we see that they have larger cerebellums, and the cerebellum is associated with keeping the body in the right posture, like balance. Balance is the word I'm looking for. And so having a brain with a large region like this for something like a shark keeps it from rolling over, keeps it stable in an optimum swimming position in the ocean. It's not like twisting in circles, spinning around. Salmon have a very large optic tectum, and so they're very visually oriented. Mammals have a very enlarged and folded cerebral cortex. So you can look at our figure over here at mammals, putting an arrow in there, huge cerebral cortex. And in fact, in humans, the cerebral cortex makes up 90% of our brains. What is the cerebral cortex associated with? Well, memory recall, language, decision making. So those are traits that are extremely accentuated in human beings, and so it makes sense that that area of the brain became so enlarged in Homo sapiens. Now this cerebral cortex is sort of parceled or divided out into about 200 distinct functional areas, each of them having densely interconnected neurons. So one of those areas, for example, is the visual cortex. And when it comes to animals like primates, where vision is so important for navigating in the trees in an arboreal environment, and vision is very important for identifying colors like fruits for food, we're talking about primates, then the visual cortex has become extremely enlarged. Whereas in something like a mouse or a rodent that has a distinct sense of smell that's much better than in a human or a dog even, then the olfactory region would become very enlarged. So the phylogenetic perspective helps us to identify what the earliest vertebrate brain looked like and what other lineages, how the brain evolved. And based on how these different kinds of creatures, animals live, we can associate the specializations in brain structures in areas with specializations in their sensory system, how they process stimuli, and how they survive. And finally here we can see in terms of the evolution of our vertebrate brains, and we're focused here on our sensory systems, especially in mammals and in us primates like humans, then the somatosensory cortex has been mapped out in the human brain by touching parts of the animal's skin, different parts of the body, and recording responses of the neurons in the brain, which neurons in the brain are responding based on which part of the skin's being touched. And so by doing these kinds of experiments, we're able to develop sensory maps, a map of the brain, understanding which part of the brain is getting signals from which part of the body. Now, areas with greater sensory perception in this figure on the top right are drawn larger. And so you see in figure A an eastern mole, and we see a naked mole rat, and we see a star-nosed mole. Now, the eastern moles are using their hands, their forelimbs, quite a bit with digging, and using their noses to smell. And so when we map out their sensory maps, we find that the areas of the brain that are most sensitive to the areas on the body by touch are indeed their forelimbs, their hands, if you will, and their whiskers and their noses. And so those areas of the body that are most sensitive to touch have areas of the brain that are sensitive to those and enlarged. Check out the naked mole rat. So they live underground in burrows and social groups, and their teeth are the primary sensory organ for sort of navigating and understanding their environment. So the teeth are drawn super large in the picture to depict the fact that in terms of what their brains are sensing on their body, the teeth are the primary target. And finally, for the star-nosed mole, it has these weird little projections of tissue coming off of their nose, and the brain mapping of the brain shows that both the front limbs and the star-shaped structure around their nose are super, super sensitive, and that's what their brain is primarily picking up. So even amongst these three kinds of moles, these three closely related species, well, they're not that close actually, we can see changes in the sensory systems, in terms of adapting for different behaviors. And finally, if we look down at humans, we can see our sensory map is showing that our fingertips and our lips are particularly sensitive parts of our bodies that our brains are wired to for sensation. And so our somatosensory cortex is highly tuned into our hands, lips, and our tongues.